Okay, so welcome everyone to the afternoon session of the second day of this wonderful awesome meeting. And so the first speaker for the session of today is Jana Ebler. She's a fourth-year physics student, and she graduated in bioinformatics from the University of Saarland in Germany. And since 2008, she joined the laboratory of Tobias Marshall at HHH University in Düsseldorf. And so she works mainly on methods for genotyping and phasing. And she's going to present, she's going to focus on genotyping with PanGenie in our talk today. So, Jana?
Okay, yeah, thank you. Yeah, exactly. So the topic of my talk is PanGenie. So this is a tool that we recently developed for genotyping. And before I start introducing the tool itself, I first explain some basic terms and definitions that I will use in the following. First of all, the goal of this method is to genotype genetic variants, and this can be SNPs in the structure variants and so on, based on short-read sequencing data of a diploid individual. And by genotyping, I mean that we want to determine whether a variant is located on both chromosomal copies of a sample, on only one of them, or whether it's not present in a sample. And
Jana, sorry, is there some noise coming out of your microphone?
Is it better now?
Yeah, that's better. Thank you.
Okay, sorry. Yeah, right. And another important term is haplotype-resolved assembly, which means to reconstruct the haplotype sequences of a sample. So basically reconstruct the two chromosomal sequences of a sample. For genotyping, there is already lots of approaches existing. And there is basically two types of approaches. The first one is mapping-based approaches, where the idea is to derive genotypes from read alignments. So the sequencing reads are first aligned to a reference genome, and then based on these alignments in a certain region of a variant, we can decide what the genotype is of the sample. The problem is that computing read alignments, of course, is quite expensive and time consuming, and that there might be a reference bias as well. The second class of approaches is k-mer-based approaches, where the idea is that we derive a genotype from k-mer counts in the reads. So the idea is if we have alleles that we want to genotype, we can define allele-specific k-mers and count these k-mers in the reads. And based on the counts of these k-mers, we can decide on a genotype. The advantage is that it's much faster compared to mapping-based approaches because we don't have to align the sequencing reads. But the problem here is that SVs especially are often located in repetitive regions of the genome, which might make it impossible to even find allele-specific k-mers because they keep repeating. So the goal of our project now is to find a method which is fast, but can also handle these repetitive or complex regions of the genome. And the idea now is that we use a k-mer-based approach, but additionally make use of haplotype-resolved assemblies in terms of a pan-genome graph in order to improve genotyping accuracy. And this is what PanGenie does. The input to the method is a directed and acyclic pan-genome graph representation, which we construct from haplotype-resolved assemblies. So the idea is we have this graph, as it's shown here in this example, where the variants are represented in terms of bubbles in the graph. And each haplotype is represented as a path through the graph, here indicated by the different colors. And for simplicity, we encode this graph structure in terms of a fully phased multi-sample VCF file, which is given as an input to PanGenie. The second input is short-read sequencing data of the sample that we would like to genotype. And obviously, that sample is not part of the graph. I mean, the sample, the haplotypes of that sample, we don't know yet. PanGenie now uses two sources of information for genotyping. The first one is the k-mer counts from the sequencing reads. And the idea is that for each bubble in the graph, we can try to find k-mers that uniquely characterize the bubble. So k-mers that only occur inside of this bubble, but nowhere else in the genome. And then we can count these k-mers in the sequencing reads. And based on the distribution of k-mer counts along the branches of a bubble, we can derive a genotype. For example, in this picture here, we can see, or if we assume that we saw all the orange k-mers in the sequencing reads of the data, but none of the gray ones, this would indicate that the genotype of the sample would be these two orange alleles in that region. But of course, this only works if we are able to determine unique k-mers. This might not be possible if we have a variant which is located in a repetitive region of the genome. And in this case, we can use these haplotype paths that we have in our graph, because we can use those to impute genotypes in regions that are poorly covered by k-mers. And we now define a hidden Markov model, which combines these two sources of information, so the k-mer counts as well as the paths, the haplotype paths. And the idea of this model is to find two haplotype paths for the unknown sample, which best explains the observed read k-mer counts, and at the same time are mosaics of the known haplotypes. And this HMM is defined as follows. For each variant bubble, we define a set of hidden states corresponding to all pairs of haplotype paths that we can select at a position. And those represent the possible genotypes that the sample could have at that position for this bubble. The emissions of the states are the k-mer counts for these unique k-mers of a bubble, and emission probabilities are based on the expected k-mer coverage that we can derive from the selected pairs of haplotypes. Transitions between the states represent recombination events between variant positions, and therefore we derive probabilities from the Lee-Stevens model to define these transition probabilities. So basically the idea is that if we have two variant positions that are far apart in the genome, the probability of switching haplotypes is high, whereas if we have two variants that are very close in the genome, the probability of switching to a different haplotype configuration is low. And this is how the probabilities in this Lee-Stevens model are defined, and this is also how we use them in our model. For genotyping, we now use the forward-backward algorithm on this model, and what it returns is probabilities for these genotypes for each bubble position. And in this way, we can then compute the likeliest genotype for each bubble. So in the end, what we have is we have a genotype for each bubble for our unknown sample. So we've now done several experiments for evaluating the accuracy of the genotyping. And this was done on haplotype-resolved assemblies of 11 samples. So we use these 11 samples, which means 22 haplotypes, in order to construct a graph and in order to do variant calling that we can, and these variants are later then used for genotyping. And in this figure here, I'm showing some statistics about the samples and about the variant calls that we observe from these samples. In panel A, we can see all the samples which are part of the graph, and we can see that they stem from various populations. Panel B shows the different substitutions that we observe for the SNP calls that we make from these 11 samples. And we can see that we have twice as many transitions as transversions, which means that we have a transversion ratio of around 2, which is a good sign. I mean, which shows that the SNP calls are actually of a good quality. And then we also looked at the length distribution of deletion and insertion alleles, which is shown here in panel C. And also here we can see that this is quite balanced, which is also a good sign. And this table here shows the total number of different variant alleles that we called from these assemblies. So we have around 12 million SNPs. Then we have around 4 million insertion and deletion events that are up to 19 base pairs long. We have around 200,000 alleles between 20 and 49 base pairs, and then we have around 150,000 SVs that we called from the assemblies. And these variants are now the basis for the experiments that I will present next. The first experiment that I present is a leave one out experiment. We did this in order to evaluate the accuracy of the genotypes that PanGenie predicts. So the idea here is that we take the variant calls and the assemblies we have for 10 samples, and the remaining sample is used for evaluation as a ground truth. And then we genotype all the variants that we have in these 10 samples using PanGenie, as well as these other genotypers that are listed here. So we use Platypus, GATK, Paragraph, GraphTyper, and Giraffe. These are all mapping-based approaches that use either the linear reference genome or a pangenome graph as a reference. And then we also ran BaseTyper, which is another k-mer-based method. And for evaluation, we use the what we call weighted genotype concordance as a metric, which determines the fraction of correct predictions for all types of ground truth genotypes, and then takes the mean. So the idea is for all ground truth genotypes that are 0, 0, we check how many of these variants would be genotyped as such by the method. And the same thing also for the heterozygous and for the homozygous genotypes. So this is what's shown here. And then we take the average. The reason why we do this is to correct for unequal numbers of ground truth genotypes, because in this setting, what we do is we genotype a very large set of variants that we observed across 11 samples, or in this case, 10 samples in a new sample, which means that most of the variants would be absent in the specific sample, which means that most of the variants would be in this category. And to correct for this, so to give equal balance on the different genotypes, we average over all these concordances. This slide now shows results. And I know there's a lot of information here, so I try to slowly explain what these plots here show. So first of all, each plot shows the on the x-axis the amount of variants that a tool was able to genotype, and the y-axis shows the genotype concordance observed for the tool. This is what I've just explained. Each tool is presented in a different color here, and we ran the genotyping on different coverages as well. So we have 5x, 10x, 20x, and 30x of coverage represented here by the different dots. And one other thing is that we, for PanGenie, had these two configurations, the blue one and the orange one. For the blue one, we considered all the genotype predictions that PanGenie made, regardless of any quality. And the orange one only considers high quality calls and treats all the other calls as untyped. Okay, and then we also consider different categories here. So we have deletion and insertion alleles. So these are all the alleles falling into diploid regions in the pangenome. And then we have these complex regions defined by all bubbles in the graph that have more than 2 branches. So all the alleles which fall into such a bubble in the graph are counted as in this complex category here. And these are the ones which are harder to genotype. And then on the top here, we show results in the non-repetitive regions of the genome. And below here, we show results in the STR, VNTR regions. Now, if we compare the genotype concordances, we can see in the non-repetitive regions that, especially for deletions, all the tools give quite good genotype concordances. So here the differences are not that big. But now, especially for insertions and also for complex variants, we see bigger differences, especially here for the SVs that are longer than 50 base pairs. We can see that the genotype concordances of PanGenie are better compared to the other ones. And this is also especially the case in these repetitive regions that are even harder to genotype. Here we can see the biggest differences in terms of performances. So this shows that that the method seems to work quite well. And also, if we look at the run times, we can see that PanGenie is much faster compared, especially to the mapping-based approaches. Also, BaseTyper, which is k-mer-based, is much faster compared to the mapping-based approaches. So this makes it especially useful in cases where we have, where we want to genotype larger cohorts. And this is also what we did in the next step. So we genotyped all the variants that we have detected across these 11 assembly samples in a subset of 100 trios from the 1000 Genomes project. And then we used a machine learning approach in order to filter the genotypes predicted for all these 300 samples. And this machine learning approach takes several features into account. One is the Mendelian consistency, genotype quality, or the genotype accuracy for the assembly samples. And the filtered set that we get in this way contained around 78% of all insertion SV alleles and 83% of all deletion SV alleles. And yeah, also the runtime for each sample was around 30 single core CPU hours. These plots now show some results for this filtered set. Panel A compares the allele frequency that we observed across the genotypes of the 100 trios to the heterozygosity. And here we can see that this follows Hardy-Weinberg equilibrium quite nicely, which of course is a good sign. There's only very few outliers. Also note that this is a log scale here. And this is also the case for insertions. Now, if we compare the allele frequencies of these variant alleles in the assemblies to the allele frequencies that we observe for the same variants across the 100 trios, we can see that also the allele frequencies seem to match quite well, which is, of course, also what we would expect. And then what we also did is we compared our assembly-based call set, here shown in red, to this database containing short read-based call sets, NOMAD. And what this shows is that there is a lot of variance in these shorter ranges here that are missed by short read-based variant callers. So there's a lot of variation which we can find when using assemblies for variant calling that we cannot find if we just use short reads. And this has also been confirmed by other studies, including the HSBC. And this clearly shows the advantage that we have when using assemblies, or in this case, pangenome graphs for variant calling and genotyping. Okay, yeah, to conclude, I've presented this haplotype-resolved assembly approach for genotyping, which we saw that it improves short read-based genotyping. We also saw that it's much faster than mapping-based genotyping approaches. And also what I just mentioned, assembly-based variant calling and genotyping allows us to access regions that previously were not accessible by short read-based calling alone. And yeah, to give an outlook, there's still lots of room for improvement. So especially genotyping rare variants that are rare in the input assemblies is quite challenging. Also, if we want to apply this method to even larger panels, there's still some work to be done to make it efficient, especially in terms of memory. And also the plan, of course, is to apply it to larger panels once they become available. So currently we're working with panels or assemblies of 90 samples. So for that, it still works very fine. But of course, in the future, we would expect to have even more. And then there's still some improvements to be done to make it efficient. And with this is the end of my talk. And I want to thank all of my collaborators, which I listed here. Thank you.
Thanks, Jana. Nice talk, very clear. I saw there are already questions in the chat, so I'm just gonna read it to you. So Gianluca is asking, what are the main problems in extending PanGenie to long reads?
So we have actually been or we have started working on using long reads as well. Currently, I think the problem is the error rate, the higher error rate of long reads, which in case we used k-mers, can of course be problematic. But our approach is now to kind of use alignments of long reads in order to improve the model, but this is still in a very early stage. But we would expect that this especially improves genotyping accuracy also in these very complicated and especially repetitive regions. So yeah, I think it's a good point also to extend it to long reads.
Sorry, I was muted. I was just reading a question from Massimiliano. I guess I should read it again. And it says, do you think using T2T genomes would be a problem for k-mer counting component of PanGenie?
I'm not sure whether I understand the question.
Massimiliano, would you like to ask directly since you're in the chat?
Yes, so the thing is the telomere to telomere genomes has this telomere component which are highly repetitive. So if those highly repetitiveness may cause problems while building like the graph and the k-mer component of PanGenie or not?
Yeah, I'm not exactly sure. It might, of course, I think repetitive regions are always a problem, especially, I think if they're represented in the graph well, then it might work, but it's obviously always challenging to genotype them correctly, especially because for these regions, as I mentioned before, usually there's not enough k-mer information, so it would be purely based on the imputation, which of course can also introduce some errors. So I think it's probably very challenging to get them right, but I don't really have any detailed results on that yet. So I unfortunately cannot really answer the question.
So Jana, there's more questions, and would you like to fix your microphone? Kind of slipped away somehow. Okay, so one question is from Zoe, I guess it's is PanGenie suitable for bacterial genomes with frequently recombinant, is PanGenie suitable for bacterial genomes where frequently recombination takes place?
So far we have only applied it to human data. One important requirement is that it only works currently only works for diploid samples. Mm-hmm. So yeah, I've never tried it for bacteria. So
okay, yeah. So there's one question from Luca Denti. There's three questions, in fact. I'll read it one by one. So first one is that, have you already seen Cage paper? If yes, do you have any comment on it? Cage, it's fast alignment, free graph-based genotyping of SNPs and short indels, and then there's a link to BioCat. So have you seen this paper already, and if yes, if you want to comment on that?
No, I haven't seen it.
You haven't seen it, okay. Just spelled Cage like K-A-G-E. Anyway, so let's go to second question. Is maybe I missed it. How did you compare the VCFs? Have you used hap.pi? If not, how did you manage comparison of complex variation, if any?
So for the comparison, the easy thing was because most of the tools are actually re-genotyping the variants so that I gave the same VCF as an input and all of the tools would predict the genotype for the exact same variant. So the representation doesn't change. And then comparing it was relatively easy because we could just match to the corresponding ground truth genotype. We've also done some comparison using VCF eval, which I think would be similar to hap.py, computing precision and recall. But what I didn't mention is that in this setting, actually computing precision and recall is not the ideal thing to do, because we are actually genotyping a very large set of variants, which can be problematic if we use precision recall, because it kind of increases the false positives. I mean, the more variants we give as an input, the more false positives we could have. And this would at some point decrease the precision. So this is why we use this different metric for evaluation.
All right, and then there's a third question from Luca. It says, maybe I missed this too, mapping-based approaches can discover novel variations. Did you consider them in your benchmarking, and how?
So for our benchmarking, we only considered the re-genotyping. So we did not consider like discovering new variants, because yeah, not all tools can do that. And this is why we skipped those variants.
But Jana, I'm curious about, like, if you have a set of reads, like a set of samples where there are variants which are not represented in the pangenome or in the VCF from the pangenome, how your tool deals with that? Like, how is this considered?
So for the variants that are like unique to the sample that we're genotyping, using a re-genotyping approach, we won't be able to genotype them correctly, because I mean, if they're not present in our input panel, then there's no way that we can re-genotype those.
So you sort of miss the private variation of the samples.
Exactly,
right?
Yeah.
Okay.
I mean, that's the issue with all the re-genotyping approaches, that like if the tool is not able to discover variants itself, then of course there's no way we can get those correctly. And as I said, we exclude these for evaluation, because none of the re-genotyping tools would be able to discover those.
Okay, cool. There's a last question from Eric. Is that what changes might be required to directly apply PanGenie to a pangenome graph? And do you see ways to handle nested variants using the models you're applying? And then he says, I'm assuming it's straightforward to get the bubble structure of the graph.
Yeah, so that's definitely one thing that we want to do to make it, use the graph directly. I mean, but yeah, it's so far, I mean, we haven't really found a good way to do it, because I think the first thing would be we would have to detect the variant region, so basically the bubbles in the graph. And also, as it was mentioned, the nested variants, there's still no way to properly handle them. Because currently what we do is we just take the whole bubble and all the paths to this bubble as alleles, but we don't really care about the nesting. But that's definitely an interesting thing. But we don't really have a solution to that yet. So that's still on the to-do list, basically.
Nice. So there's someone typing right now, Alex. If you fast, we can ask. Okay, kind of not typing anymore. So I didn't see any other question in the chat. So I just want to say thank you, Jana. That was very nice talk, very clear.